This Week in Virology, the podcast about viruses, the kind that make you sick. You're listening to This Week in Virology. This is a special episode recorded on September 5th, 2012. I'm Vincent Racaniello, and with me is Professor Ian Lipkin. Thanks for joining me today, Ian.
A pleasure.
Today we're here to talk about a paper that will be published on the 18th of September. And the title of this, the journal is MBio. The title is A Multicenter Blinded Analysis Indicates No Association Between Chronic Fatigue Syndrome / myalgic encephalomyelitis and either xenotropic murine leukemia virus, related virus, or polytropic murine leukemia virus. And let me read just the last names because I think everyone should know who's on this paper. Alter, Mykovitz, Switzer, Ruschetti, Lowe, Klimas, Komarov, Montoya, Bateman, Levine, Peterson, Levin, Hansen, Genfi, Bat, Zhang, Wang, Li, Hung, Li, Samarov, Henine, Coffin, Hornig, and Lipkin. It's not probably the biggest author paper you've ever had, right?
It's close.
It's close. So this is the culmination of a study that you've been coordinating for over a year now. Is that right?
It's 2 years.
And it began in 2009 with a report that Xmrv, a new retrovirus, might be involved with chronic fatigue syndrome. And it's been quite a ride since then. There have been a few positive studies, but mostly negative ones. So can you tell us why was this study undertaken that you have coordinated?
There are two issues in pathogen dediscovery. One, of course, is to persuade the scientific community that you've done everything necessary to address the question, whether or not you've made a substantive link or not. And the other is to do everything you can for the community that you're trying to serve, which is the patients, the families of those who are afflicted with the disease. And although many in the community have felt for a long time that this issue was settled, the many members of the community have not felt that to be the case. And the people who participated in this study, who were critical in generating the data that led to the whole Xmrv polytropic murine leukemia virus link to the chronic fatigue syndrome, myalgic encephalomyelitis, were Harvey Alter, Judy Mykovitz, Frank Ruschetti, Shai-Ching Lowe, and of course, the people who work with them. Now, although some would say that these individuals retracted their papers, when you talk to them, they did not, in fact, retract those papers. They will tell you that they were pressured by journals to retract, to participate in retraction. So while the journals may have retracted the papers, the authors did not. And until the authors have a chance to go over the original information, the original assays, to try to test these with well-characterized samples, and then find the hypothesis wanting, they're not going to be willing to come out in public and say, we made an error, it was an honest mistake, but we find no evidence for this link between these viruses and this disease. And this is why, in fact, this study was undertaken. It was undertaken with very strong support, even after some of the negative papers began to appear from Tony Fauci, Francis Collins, Tom Frieden, and Harold Varmus, because they realized it was critical that we address this in a forthright, coherent, comprehensive, indisputable fashion. So what we set out to do was to identify cases using criteria that were established by the laboratory people as well as the best clinicians in the field. They were collected over a wide geographic area so that if there were differences in the causes of chronic fatigue syndrome in one part of the United States or another, we would not be confounded by just looking at one area. This is very important. We made certain that the controls we selected were matched well, and we collected the samples that the investigators responsible for original work wanted us to collect. We coded them. We sent those samples to them. We allowed them to use whatever assays they wanted to use. The only thing we insisted upon was that the criteria for calling a sample positive or negative had to be set up in advance, and that once they were established, they could not change. And we then allowed them to proceed. We found, they found what they found. And once we collected all those data, if there was any sort of discrepancy from one sample to another because they received more than one sample, we tried to reconcile them. And at the end of the day, what we found, in fact, was that although the laboratories were able to recognize the positive controls, those were provided to us by John Coffin, there was no association between chronic fatigue syndrome, or myalgic encephalitis if you prefer, and either of these two viruses. No one found any sequences by PCR. The serology was a little more tricky because we don't really have positive controls for it, and the results were somewhat discordant. But again, there was no association between the presence of antibodies as described by Frank Raschetti and status of the case. So when all these data were assembled, we met in teleconference, and everyone agreed that the hypothesis had been tested and found not to hold up. And I will say that, in deep appreciation of for the sincerity of all the investigators, everyone held to their word. So they are speaking out and saying, we feel that this study was fairly done. We feel that the hypothesis has been tested, and we find no association between the viruses that we previously reported and this disease. Now, I've been involved with other sorts of studies where we've tried to look at the role of infections in chronic diseases, ALS and enteroviruses, Borna disease and chronic fatigue syndrome, measles virus and MMR and autism. And I've never had an opportunity before to get as many people involved so that when we're finished, there's absolutely no question but that we've tested it and found that there's no link. And I'm hopeful that this is going to settle the issue once and for all. There's been a side benefit to this whole process, and that is we've established a working group of people who are capable of collecting materials. We've been able to store sera and pbmc so that other investigators who want to do either microbiology or genetics or proteomics will be able to access those samples through the NIH. There will be calls for proposals, and there will actually be funding associated with that. During the interim, we offered the laboratories and the investigators and the clinical sites who are engaged with this work to put forward proposals to do something with these materials in advance of that RFA, though without any funding. And two of those groups, several groups applied. Two of them were selected by the entire team, and they will be receiving plasma samples that they can then study. The primary focus has been on microbiology and genetics, but I'm sure there will be other applications as well. So we've been able to hold back as much of the sample as possible. So I know some in the community, the scientific community, feel that this was not, money well spent. But the fact is, I think that we've obviated a lot of, missteps that might have followed with clinical trials and such for antiretrovirals. And in addition, we've been able to establish a sample bank that will be helpful for years to come. And we've been able to hold, I think, the attention of the community.
So the samples that are going to be available are the samples collected as part of this study?
That's correct.
That's 147 case patients, 146 matched controls.
That's correct.
Right? And you have enough material for many years of investigation.
Well, I remember, depends on how greedy people are, as usual. But there are at least 15 pbmc samples. Now, those are difficult to aliquot, each with 1 million cells. And so those will go to individual projects. The plasma, however, we can aliquot, and for people who want to do proteomics or do microbiology using these plasma samples, we have enough for probably 50 laboratories.
So tell me how these samples were selected. You mentioned that all the investigators were given the opportunity to participate in that. So did each group have X number of patients for this study?
Yeah, so
how did that work?
So we initially did a power calculation, trying to figure out how many samples we needed, based on the worst and best case scenario from the Mykovitz paper and the Low paper, so we could determine the minimum set. And based on those calculations, we sought 150 subjects with disease and 150 who were match controls. Now, we brought in more than that, obviously, because as we began to examine these people and they met clinical criteria and we collected their blood, we also sent those bloods off before deciding to include them to a clinical laboratory here at Columbia, and we used other laboratories as well, commercial laboratories as well, to make sure that they didn't have hypothyroidism or some sort of cryptic hepatitis or anything that might confound the diagnosis of chronic fatigue syndrome. And so we initially, given we had 6 sites across the United States, the notion was that each one was going to get 20 to 25. At the end of the day, some were more successful in recruiting than others were. But on average, we had somewhere between 20 and 30 patients selected per site and with match controls. Now, the laboratories, so the clinicians met and the laboratorians met. Everyone decided and again, agreed on what the criteria that would be used, should be. We used the most stringent criteria possible. So we selected people who met the Canadian criteria. People who they also had to meet the Fukuda criteria. We also decided that we wanted to select people who had a prodrome that was consistent with an infectious disease. So fever, night sweats, lymphadenopathy, swollen lymph nodes, anything suggestive of an infection, so that we would give the hypothesis the best possible chance of success, which has not been done in the past, so that there would be no way to go back a priori and criticize the study design and say, if you'd looked at this population, you would have found Xmrv or PMLV. And it's critical that you do that. If you just go into a freezer and try to pull out cases that have been previously called positive, you can always say, well, maybe there was a thaw, maybe the titer dropped, who knows what could have gone wrong. In this instance, everything was pristine, from the selection of the cases, the criteria that were used, the processing, the materials. So the samples were sent to us overnight. We purified pbmc using, standard classical methods. Everything was stored in freezers here, so there was no possibility for contamination. And we have had no PMLV or Xmrv in our laboratories. And all the assembly we do is done in positive pressure conditions, so we don't have to worry about contamination of materials. And in fact, we didn't have any evidence of contamination. So we set it up again so that people could do what they wanted to do. Now, this means that some labs did things that other people didn't do. So the FDA analyzed pbmc as well as plasma, as did the Mykovitz, Racchetti, Hansen laboratories, and the plasma was really only analyzed alone was analyzed at the CDC. Now, during the course of this study, lots of things happened that made it very difficult to continue. And I think it was Harold Varmus who said, this is worth a paper in itself, or maybe a movie.
Didn't you do that already?
So, as you may recall, Judy Mykovitz had difficulties in the WPI. The WPI, which stands for the Whittemore-Peterson Institute. The P in WPI stands for Peterson, who doesn't talk to the Whittemores anymore, so that's another story in itself.
He's on this paper, right?
Yes, he's on this paper as well. He was one of the original people who, with Paul Cheney, described this group in Incline Village, where this syndrome was early to be reported. And then Judy Mykovitz, wound up afoul of the WPI, and she wound up in jail, which was quite remarkable because I wound up involved with those negotiations, trying to broker an agreement between her lawyers and Judy Mykovitz and the Whittemores to get her out of jail. I mean, essentially what happened was when it began to look as though the hypothesis was unraveling, people began to push, the onus of responsibility onto Mykovitz for that work. And of course, she wanted to have access to her lab notebooks so that she could at least describe what she'd done and relate it to the individuals there. She took those lab notebooks, and I had conversations with Harvey Whittemore about whether or not she should have access to her lab notebooks. And I would say, I understand that you want to keep the original lab notebooks, but every scientist is entitled to have copies of his or her records. How else can they talk about what they've done, replicate their work, provide detailed protocols, and so forth? Well, that didn't rest very well. She was accused of stealing intellectual property. Well, if the hypothesis doesn't hold up, where's the intellectual property? So there were all these things that were circling, and then we had to find a way for her to be engaged in the study. So Maureen Hansen at Cornell agreed to receive samples processed by Frank Raschetti. Harold Varmus agreed to allow Frank Raschetti to continue to participate in the study and to receive samples and characterize them. And at one point, the CDC said, we don't really have the time and energy to invest in this. And I appealed to Tom Frieden, and I said, look, we need to close this loop once and for all, so that we don't have another situation with MMR and autism, which lingered, as you know, for more than a decade. And we still have people who are reluctant to vaccinate their children with MMR, despite the fact that, that's been clearly laid to rest. So everybody got on board, pushed this very strongly, and this is the result.
So how did Judy participate in this?
So
in all of this?
So Judy, while Judy was in jail, and after Judy got out of jail, she was in contact regularly with Frank Raschetti and with Maureen Hansen, and was responsible for the study design and for making decisions about how you call a sample positive or negative, and reviewed the data and participated in writing the paper. There was a lot of negotiation in the drafting of this paper, as you might imagine, because it's very sensitive.
Sure.
But everybody signed off on this. We went through literally 50 different iterations of the paper, and after it was signed off on by all the authors, then it first had to go up the chain at the NIH and the CDC, so that they could approve it as well.
Do you have the original Word document with all the changes embedded in it? That might be a historical document.
Yeah, the problem is it crashes.
It's too big.
It's too big for Mac.
So none of the samples used were samples from any other previous study. Is that correct?
That's true. These are all, well, I can't say any previous study, but any previous study in which these people have been engaged.
Right. Okay.
Yeah.
And the positive cases, the individuals diagnosed with CFS, were these individuals who had the disease for varying lengths of time, short, medium, long?
Yeah, so in the paper we spent a fair amount of time talking about the nature of the disease. One of the challenges, of course, with a disease like this one is that because it's called chronic fatigue syndrome, you can't get there during the acute period, during the acute ictus. So, but the mean age, at illness onset was 35 plus or minus 10 years. The mean duration was about 16 years plus or minus 8.5. So there isn't anybody here, it's not like we were able to get people 6 months after the onset of illness, which would be the closest you could be, and still have a diagnosis of chronic fatigue syndrome. And I don't really see any way in which such cases are going to be found.
Right. Now, you did one of the things that was done. So, or let me back up a bit. You received blood samples in your lab, is that correct? That's
correct. So the patients would be seen, bloods would be drawn, they would be sent to us.
Is they drawn somewhere else?
Yes, the bloods are all drawn in commercial, in a commercial phlebotomy suite.
Okay.
Then they're shipped to us overnight. They're received, which means we're receiving 5 days a week, because they also have clinic on Fridays. and we would then process them, and we would aliquot them, and we would code them with barcodes, and then we would send off a sample of plasma to clinical laboratories to test for things that we wanted to know about. And this hasn't been done before in the past either, but we looked at a wide range of parameters that we thought might be important as exclusionary diagnoses. Now, I've mentioned some of these already, like evidence of liver disease,
Right.
and so forth. But
Other
we looked
viruses.
at, but Table 1 has a whole series of different things that we examined that are important, and you can look at all the various exclusionary criteria. So once those laboratory values came back, we then decided which of the samples would be taken and would be submitted to the individual labs for testing.
So what was the cut down from the original samples to what was actually tested for viral sequences, the 147, 146?
I don't remember the total, but that's how we got down to 147
All right.
and 146. It wasn't as though we drew 200, for example, because a lot of these cases were excluded at an earlier time point because they simply didn't meet various criteria.
Right. So one of the tests so then you sent these coded samples out to the various labs, and then you said they could do what they wanted with them, assay them in
That's
the ways
correct.
that they wanted. And one of the ways that CDC and FDA did was to do PCR on plasma or pbmc, right?
Correct.
So in some cases, this was RT-PCR. They would
That's correct.
They would produce RNA from the samples, reverse transcribe it.
Mm-hmm.
What kind of primers were used for the PCR? Was this agreed upon by everyone?
No, that's a critical point, and I appreciate your bringing that up. For the purposes of this study design, I really don't care what primers people use, because if I ask you, and you've previously reported something as being positive, to use my primers, and you don't get a finding that correlates with what you previously reported, you'll simply tell me that this is because I forced you to use my primers. So we wanted to circumvent that criticism. So the one thing we did do is we said to people, whatever primers you want to use, whatever reagents you want to use, obviously the point of collection, we can't vary that. That's entirely up to you. You tell us where you want to purchase these primers, and we will pay for them. So what we then did is we actually arranged for various commercial suppliers to provide reagents to people who needed them. But they were the reagents that they designated, and they list them, and you can obviously look up what they've used. And these differ from laboratory to laboratory. Now, we did ask that people go back and sequence their products and see what they found, but that's not terribly informative. What's really critical, obviously, is the end result, and that is to say, can you confirm or refute the finding of the relationship between the viruses and the disease? And that's where, of course, nothing was found, right?
So the CDC and FDA both did RT-PCR of plasma, so that's looking for virus, correct?
That's correct. Or I don't know that
RNA sequences.
Nobody was doing a viral purification protocol.
Right.
So it's not like they did a nuclease treatment and then extracted. They were just taking plasma and doing traditional extraction. The FDA wanted to access pbmc. Now, as you may recollect from the early days of the response to the criticism of the Mykovitz paper, what Mykovitz-Ricchetti said was that the key was the growth of these cells,
right,
which then allowed expression of virus. So we provided the reagents that were required to culture the pbmc so that they could test that hypothesis. That didn't make any difference. That didn't, but it could conceivably have done so, but it didn't.
Right.
So those were also negative.
So that was PCR of cultured pbmc.
That is correct.
This is not RT-PCR. This is extracting DNA.
Yes,
correct.
But again, the key issue here is the culture. So
yeah,
the FDA did not culture their cells, and this is one of the reasons why it took so long to get through this. Study is because you have to be able to culture these cells,
Sure.
and sometimes Frank Racchetti found that he wasn't able to propagate the cells. So we had to send him another aliquot, and he had to try again. So that then takes several weeks. And then after that was done, the PCR was done, whether it was plasma or PBMC, all the PCR was done for that group at Maureen Hansen's laboratory up at Cornell.
So we looked for viral RNA or DNA, which would be integrated DNA, presumably,
Correct.
Right? All negative on the positive cases, the CFS-ME cases, which are 147. None were positive in any case for PCR or RT-PCR.
Correct.
And the same for the controls, which is 146, 0 for all of the RT-PCRs and PCRs, including the cultured PBMCs. Right? So that's CDC, FDA, and Mykovitz, Racchetti, and Hansen. So that's resoundingly negative.
So the only issue is the plasma serology.
Right.
And
so what was done there? Tell us how that's done.
Well, Racchetti has a proprietary assay that he uses for test for antibodies reactive with cells. And as you see, there was no difference between cases and controls. Controls 6.2%, cases 6.1%. And because we don't have a positive control, I really don't know what that means. Now, many, many years ago, when I was asked by CDC to look into the potential role of Bornavirus in chronic fatigue syndrome. So this was back in the mid-1990s to late 1990s. There was a report that came out of Japan suggesting that Bornavirus was implicated. And 50% of cases were reported to have nucleic acids, and there was a family found where 3/4 of the people in the family had Bornavirus nucleic acids and were also antibody positive. So Brian Mahi was then director of DVRDD, who ran afoul, as you may remember, of this whole issue of whether or not enough money was being invested in this research, asked me to look into this, and I looked into Gulf War syndrome, which was also very topical at the time, and I looked at these chronic fatigue syndrome patients, many of them coming out of a clinic that was run in the Karolinska, which at that point was one of the best clinics for CFS in the world. And we found no evidence of Bornavirus nucleic acids. And our ELISAs were positive, but our Western blots were negative. And when I tried to report this finding, I wound up going to progressively less impressive journals until I finally wound up at the one in which it was published. And there was a great deal of reluctance to publish the paper because the saying at that point was, you're just not good enough at doing this, and this was why it's all negative. And it took almost 2 years to publish that particular paper, which is how I learned the lesson of making certain that the people who originally do the report are the ones who are engaged in proving or disproving the concept. But at that point, the one thing that struck me was that many of these individuals, I think it was close to 2/3, something like 67%, were reactive with Bornavirus proteins and ELISA, but negative in Western blot. But they were also reactive with FLAG and beta-galactosidase, and I was left to conclude that they had polyclonal B-cell activation. So I at the very end of this paper, which appears in something called Journal of Neurovirology, I said, these patients are clearly sick in some way. They have some immunological activation. I don't know why, but it's not Bornavirus. So in this instance, when you compare the serology between the cases and controls, you don't find that discordance. You don't find more reactivity in the CFS patients than you do in the controls. But I am convinced, after working in this field for a very long time, that this is a bona fide syndrome. I don't believe that it necessarily has a single cause. I don't pretend to have insights into the pathogenesis of CFS. But one of the things that we want the community to take away very clearly is that the people who have been engaged in this study and many others around the world are committed to trying to understand why they're ill, We have reagents, we have resources now that can be applied to this task, and that just because this particular hypothesis hasn't borne fruit does not mean that there won't be an answer downstream. And we're trying to do that work as rapidly and efficiently and as rigorously as we can. Now, one of the other things that I think is important for your listeners to take away from this particular investigation is that we need to have rules for interpreting data. We need algorithms by which, when people have a tantalizing finding, it can be tested. And we've got Koch's postulates, and people use Koch's postulates to this day, but they don't serve us very well in this era when we don't have an animal model for a disease and we don't have the ability to grow the organism necessarily. So I have another paper that's under review right now. It's not on chronic fatigue syndrome, but it's specifically about the issues of how you talk about levels of certainty, levels of confidence in the association between a finding and a consequence. And I hope to be able to talk with you about that at a later time.
What kind of rules are you thinking about?
Well, I have 3 levels.
Mm-hmm.
The lowest level is the phenomenon. You find something but it doesn't tell you necessarily that there's
Right.
any linkage. And then the next level talks about finding something in association with an organ that's affected.
Mm-hmm.
It looks at clusters of disease where you find something. It examines a burden. It talks about biological plausibility based upon analogies to other systems. So, for example, this virus causes cancers in cows. It could equally well cause cancers in humans. A papilloma virus, sort of an example. And the last level, the most compelling level, of course, which I call level 3, is where you either complete Koch's postulates or you demonstrate that a very specific drug can eliminate disease, or you demonstrate that by vaccination, the incidence of disease drops dramatically. Of course, Koch's postulates is the gold standard, but I don't think it's essential.
How, in this case, the Xmrv CFS story, how did we not follow those rules?
Well, I think with the initial publication, we had an association. We had some epidemiological data that suggested that there were differences between cases and controls. But that can be that you can be confounded. Let me give you a couple of examples. There was one particularly poignant one a few years ago where someone thought they had found the cause of a disease. I don't want to go into more details because I don't want to embarrass these people. But in essence, it was a respiratory disease. It was a disease of children, and unfortunately, the samples were collected during an outbreak of a respiratory disease in a given geographic location, and the controls came from during a different time of the year. And as a result, there was this respiratory pathogen that was passing through the population. It had nothing to do with the disease in question. So you probably know what I'm talking about. But that's why we decided to select all of the cases within a certain time frame so that we could be certain that we weren't being just simply looking at an outbreak that was passing through a community. And in addition, this was the reason why we decided to go for a geographic diversity as well.
Right. So the virus Xmrv was originally identified in a screen of prostate tumors. And although your paper has nothing to do with prostate cancer, there's still some individuals who believe that there may be an etiologic role for this or a related virus in that disease. Any thoughts on that?
Well, I agree. And according to, the strategy that I would pursue in this sort of work, I would suggest, if you really wanted to address that rigorously, though I'm not eager to jump back into the world of Xmrv, and I don't intend to. As I explained, I've done my bit for NIAID for the year in this respect. I think what would be required would be the people who originally published that work should be sent blinded samples collected from individuals with disease, and they should be asked to test them and report their findings. But until the original group that makes the report withdraws the finding based either because they say we made an error that we're admitting to, or they retest new samples and say we've done the best we can to approach this problem rigorously, that question is going to be open.
Right.
And I think that's unfortunate, and it's a disservice to patients who are continuing to pursue this hypothesis. It is imperative that we find ways to rapidly address and reconcile these kinds of findings with other data so that we can move on.
Well, it seems that requires people to work together, and you were sort of a third party in this case, so you were able to get this to work, and it's probably why NIAID came to you in the first place. But when you have people having different findings, it's hard to get them to talk to each other. So what do? You call them up and say, hey.
How do you get to yes? It's very difficult. I find that breaking bread with a little bit of alcohol is quite helpful.
Yes. What if you don't drink? Break the bread.
It's even better.
So you mentioned just a few minutes ago, you hope this is your last association with Xmrv. Yet, it sounds like you're interested in CFS ME. So what's the future with that?
I am indeed. So what we're doing is we have two efforts underway. First, with the Hutchins Family Foundation, we've received some support to pursue pathogen discovery and biomarker discovery in people with CFS-ME. And what we've done is to go to all of these clinicians and others who have previously found agents using commercial laboratories and have constructed a mass state PCR system that allows us to rapidly look at their leading candidates. And in addition, we're going to be pursuing high-throughput sequencing on the Illumina platform. We're also doing RNA-seq. We're looking for RNA markers. We're looking for linked RNAs, any other sorts of insights we might have that would enable us to understand the pathogenesis of the disease or develop markers that could be used for diagnosing people or making predictions about who's likely to respond. We're also have a large proteomics project associated with this. So, and I am, completely unbiased with respect to what we find. So we're using unbiased high-throughput sequencing, proteomics, same sort of thing. It really is a discovery platform.
What about the microbiome? Could this have some role in the disease? And it should be
so
looked at?
The microbiome, unfortunately, in this project, we were not allowed resources to do those kinds of collections, which I think is something that needs to be done. Under the auspices of the Hutchins Family Foundation, we are indeed collecting rectal swabs, so we will be able to look at those types of materials.
The other issue that, as you know, there are a lot of individuals who have followed this story, many with the disease. Many of them accept or will accept the conclusion and hope to move on, but there will be a vocal cadre of them who don't buy it. And some of them will say, well, you haven't looked in the right place. Why pbmcs? How do you know that is where the action is?
Well, that's a good point, and that's another reason for doing this study as we did. We gave these investigators an opportunity to specify what samples they wanted. And they wanted pbmc, and they wanted plasma. And they wanted an opportunity to culture the pbmc. So when you start a study like this, you have to have ground rules. The ground rules that I insisted on are, the criteria
Right.
for case selection, for sample collection, for sample processing, for analysis, for reporting, have to be established in advance. And that's the only control that I'm applying from the outside. So you can never exclude the possibility that had we looked someplace else, we would have found something different. But given that the original reports were based upon similar types of samples, similar types of analyses, these individuals felt comfortable with saying, we believe that this study was fair. So we have a press conference, on on the 18th, where Harvey Alter, Judy Mykovitz, Frank Raschetti will be present, and they will have an opportunity to answer questions from the press. And based on my understanding of what we've talked about, there won't be any surprises. They're going to say, we put our full weight of enthusiasm, belief, confidence in what was done here, and we feel that we were given a fair shake. Excuse me. And that I think it's time to move on.
In fact, this podcast will be released on the 18th, and I have this is your press release, which shows that what you're saying is correct. Everyone seems to be agreeing here. And I should read the last
quote from Mykovitz.
Yes. Do you mind if I read that? Since
Not at all.
It will have been spoken.
Not at all.
Although the once promising Xmrv and MLV hypotheses have been...
Start higher than that.
You want me to go higher?
I greatly appreciated the opportunity.
I greatly appreciated the opportunity to fully participate in this unprecedented study. Unprecedented because of the level of collaboration, the integrity of the investigators, and the commitment of the NIH to provide its considerable resources to the CFS community for this important study. Although I am disappointed that we found no association of Xmrv/pMLV to CFS, the silver lining is that our 2009 science report resulted in global awareness of this crippling disease and has sparked new interest in CFS research. I am dedicated to continuing to work with leaders in the field of pathogen discovery in the effort to determine the etiologic agent for CFS. Yes, the next quote is yours.
Well, that's okay. I mean,
we know what you think.
The important quote is hers, and
Yes.
I think this takes an enormous amount of courage and class.
It always does to admit you're wrong. But that's the key, right? That's the key to moving forward. And that's good to hear. And, of course, in your paper, the discussion begins, our results definitively indicate that there is no relationship between CFS/ME and infection with either Xmrv or pMLV. And that is, everyone signed off on that, you say?
They've all signed off on it. Everyone has read the press release.
I would agree with Xmrv for sure, and pMLV as far as you've shown here, but maybe somewhere else in the body there's another virus that is causing this, or some pathogen. We're not ruling that out, and I don't think you said that.
I am heavily invested, personally and professionally, in that search.
And when you find
Not personally, but professionally.
Professionally, when you find it, we'll have you here to talk about it. Anything else you'd like to say before we wrap up?
Thank you.
Thank you for coming by. I appreciate it. So the press conference is going to be live streamed, I understand?
Indeed.
And we will put a link at virology.ws and TWiV.tv so that everyone can find it and listen to it as well on the day of the 18th. Ian Lipkin is a professor in the Center for Infection and Immunity at the Mailman School of Public Health at Columbia University. Did I get everything right there?
Perfect.
Thanks for joining me today.
Pleasure.
I'm Vincent Racaniello, and you can find me at virology.ws. You've been listening to a special episode of This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.